A very good morning to all. I hope I'm heard. Okay, welcome to the 23rd episode of Breakfast at UMHealth webinar series. I'm Ivy Chung from the Department of Pharmacology, and I will be your moderator today. It is really a great pleasure to have two distinguished speakers gracing our breakfast at UMHealth. We shall have our Q&A at the end of the webinar, so please write your questions in the Q&A box. First, I would like to introduce our first speaker, which is Professor Dr. Mohammed Abdullah Alshawsh. Professor Mohammed has been with the Department of Pharmacology for more than 10 years, and only just recently, he has pursued his research career at Monash University, Australia, serving in the School of Clinical Sciences, Faculty of Medicine. His research focuses on investigating the pharmacological effects of flavonoids, polyphenols, and also probiotics in colorectal cancer and metabolic syndrome. He has extensive experience in systematic review, meta-analysis, microbiome, metagenomics, and toxicity testing. He has been very excited about this event. However, he has some religious commitment because today the Victoria State is observing the Eid al-Adha. So we will play a recorded talk prepared by Professor Mohammed with the title Metagenomic Analysis of the Gut Microbiota, Insights into Colorectal Cancer Prevention. Secretary, you can please start the recording. Thank you.
Assalamu alaikum and very good morning. I'd like first to thank Prof. Ivy and other team members for organizing this webinar. And also I'd like to apologize for not being able to join you live today, as Wednesday is Eid celebration here in Melbourne. And instead I have recorded this video. So my presentation or talk today about the metagenomic analysis of the gut microbiota and its role in colorectal cancer prevention. I will start with a brief introduction about the gut microbiota. So nowadays, the gut microbiota considered as a complicated organs. And just to imagine how huge this microbiome, which is the collective genome of the whole gut microbiota, it's almost 100 times more than our own genome, the whole genome of all of our cells. And this gut microbiota, it contains several microorganisms, bacteria, fungi, archaea, and viruses. The relationship between the gut microbiota, all those microbiome, and gastrointestinal disorders or diseases, including ulcerative colitis or irritable bowel disease, and even including colorectal cancer, it's considered complex, and until today, it's not really fully understood. On one hand, there are certain microbes, they are associated with higher risk of colorectal cancer and ulcerative colitis and other gastrointestinal diseases. While on the other hand, there are certain microorganisms, they play a protective role and they prevent the development of colorectal cancer. Interesting clinical study published a few years back, they show or revealed that the makeup of the gut microbiota of patients with colorectal cancer are really completely different compared to the gut microbiota makeup of healthy individuals. These findings are really interesting findings. In this table, you can see few examples of taxonomic gut microbiota composition in the gut of healthy individuals. The most dominant phylum in our gut, mostly it's Firmicutes and then Bacteroidetes. At the species level, those highlighted in red color are microbes associated with higher risk of colorectal cancer, especially Fusobacterium nucleatum and also certain species of Escherichia coli, H. pylori. Also, there are few studies show that H. pylori associated with higher risk of cancer. While those in brown color are species based on research associated with preventive role against colorectal cancer, especially Faecalibacterium longum and Faecalibacterium bifidum, also Faecalibacterium brusnesi, Lactobacillus, certain strains of Lactobacillus, and then Akkermansia and other strains as well. So, as I just mentioned, certain strains, it's associated with higher risk of CRC or colorectal cancer, so it's considered as carcinogenic. The most well-known microorganism is Fusobacterium nucleatum, and the mechanism how this microbiome it involves or it considered as a risk of colorectal cancer by inducing chronic inflammation, by interfering with the immune system, and also sometimes it produces different types of toxins that it can directly damage the colon cells and promote the cancer. Here you can find a list of other microorganisms or microbes that are associated with higher risk of cancer, like Bacteroides fregalis. It produces certain toxins called Bacteroides fregalis toxin, which will damage the colon cells and promote inflammation and cancer. Streptococcus gallilolyticus, it induces also chronic inflammation. There are certain species of Escherichia coli, especially enterotoxigenic Escherichia coli, which produce toxin that can contribute to cancer development. The other pathogenic Escherichia coli called enteropathogenic Escherichia coli, also it promotes the inflammation. It does not produce toxins, but it considered as risk for colorectal cancer by promoting inflammation and damaging the colon cells. H. pylori, based on previous studies, it induced the chronic inflammation. On the other hand, there are, as I mentioned, several strains and species associated with preventive role against colorectal cancer. The well-known Faecalibacterium, especially Faecalibacterium longum and Faecalibacterium bifidum, they help to prevent CRC by promoting a healthy gut environment and by reducing the inflammation. Lactobacillus, another well-known good species that it inhibits the growth of cancer cells and it promotes the healthy gut. Faecalibacterium brusnesi, and I will present some of the our team finding about this probiotic. It has, based on the previous study, it has anti-inflammatory properties which prevent colorectal. It could involve in preventing CRC. Akkermansia, it improves the gut barrier function, it reduces the inflammation. Other strain, which is Roseberia intestinalis, it produces certain short-chain fatty acids like butyrate, which it exert anti-inflammatory properties. So after this brief introduction about the gut microbiota, let's move to the metagenomics. So what is the metagenomics? It's a study of genetic materials of the collective microorganism recovered directly from either clinical samples like oral or gut, or from other environment like soils. And the method that we use it to analyze this metagenomics called sequencing. Before introducing the term metagenomics, which was introduced in 1998, we used to, the only method is to culture the microorganisms. If we want to identify what types of microbes in our gut, the only method is to cultivate those microbes in lab. but unfortunately, only less than 1% of those microbes can be effectively cultured. So since the modern genomic techniques introduced, it really changed the way how we diagnose and identify the microorganism, either in gut microbiota or other environment. So far, there are more than 1 million genes were sequenced and deposited in public databases. So the question now, why we study the microbes, the microbiome? Why we need to study the microbiome? So the microbiome, it's the collective genome of the gut microbiota or the collective genome of the oral microbiota or skin microbiota. There are several reasons and benefits. One of them is to investigate how we can protect ourselves from pathogens like Clostridium difficile. Also for studies about the antibiotic resistance, for anti-cancer efficacy or cancer prevention studies. Also the microbiome and metagenomics are important. For certain studies about the absorption and production of nutrients by the gut microbiome, gut microbiota, especially in obesity and type 2 diabetes, and also for regulation of immune system, since gut microbiota also involves in regulation of immune system, especially in certain chronic disease like irritable bowel disease and other autoimmune diseases. So also microbiome studying or microbiome is essential here. Gut microbiome also, it interferes with the drug efficacy and the field of study that it deals with the relationship between the gut microbiota and drug efficacy and absorbance and metabolism, called pharmacomicrobiomics. And we have published, our team actually published an article in FASAP journal about the influence of gut microbiota on drug and xenobiotic metabolism. Feel free to go through this interesting articles. So what are the most common approaches for metagenomic analysis for the gut microbiota? The first approach is 16 RNA sequencing, while the second approach is shotgun whole genome sequencing. For the 16S RNA, it's mostly for taxonomy. So the 16S RNA is not able to produce functional finding or information. If we need to have functional study, we have to use shotgun whole genome sequencing. For 16S RNA, it uses variable regions, especially V3 and V4, because those regions are hyper variable and they differentiate different one genus from another. So we use those variable regions to distinguish between the genus. The 16S RNA is not able to distinguish at the species level. So it can classify the microorganism based on the phylum up to the genus, but is not able to classify or identify the microorganism at the species level. And of course, there is no functional information from the 16S RNA, but the good things, it has less complex analysis, and it's rapid and it's cost effective. Nowadays, it's cheaper compared to shotgun. On the other hand, the shotgun sequencing, it sequences the whole DNA. So here, it sequences only certain parts of the 16S RNA, which is only those variable regions, V3 and V4, while the shotgun, it sequences the whole DNA. It produces, of course, more information, including functional studies. But one of the challenges, it's the complex pipelines and the complex data analysis, and also it has higher cost. And there is issue of contamination. So if there is any fragments from the host genome it will affect, absolutely, it will affect the result. So to differentiate between those two techniques, if you can imagine, if there is a puzzle, the 16S RNA, it will identify maybe the frame or the corner of those puzzle, while the shotgun sequencing, it will identify or show the whole picture of the puzzle. But the 16S RNA, it's good enough to classify or distinguish the microorganism until the genus level. Let me just check the time. Okay, so this is the analysis by Blyans for both 16S RNA and shotgun. So there are two methods for 16S RNA, either OTU, which is operational taxonomic units, or ASV, which is umbilicon sequence variants. They are more or less the same. They produce similar finding. In the past, mostly they use OTU. Nowadays, they started many researchers to go for umbilicon sequence variant. So the 16S RNA, it produces taxonomic distribution of the microbes or microorganisms, and it shows the diversity and richness of the microorganism. So it produces OTU clustering. While for shotgun, in addition to the taxonomy distribution and richness, it provides some functional studies or functional findings. Let's start with the 16S RNA sequencing. 16S RNA gene, it's considered as taxonomic genomic marker that this gene, the 16S RNA, is relatively short, and it's common in all bacteria, archaea, and fungi. But for 16S RNA for bacteria and archaea, but for fungi, it's 18S RNA. And the 16S RNA is considered the most commonly used molecular marker to assess both diversity and also the abundance of the gut microbiota or other microorganisms on the skin or oral or whatever, or soil. So by amplifying those highly conserved genes, either 16S RNA for prokaryotic or 18S RNA for eukaryotic, we are able to classify the microorganism. And as I mentioned, the primary targeting for the primers, it's the hypervariable regions, V3 and V4, because those are, it can be used to differentiate the microorganism. Amplification of 16S RNA sequencing, it's limited, as I mentioned, by mainly gene fragments rather than the whole length of the genes. Shotgun sequencing, it does that. It sequences the full length of the genes. And usually, once the organism display between 97 to 98, we can identify this gene to be part under the same OTU. So when we cluster the reads, we classify them under the same OTU when they display similarity of more than 97%. This illustration shows the sequence of the work. So after we collect the samples from fecal materials or whatever, but today I'll focus more on the gut microbiota, so fecal material. The first step is to extract the DNA, and then after that, we do amplification, 16S RNA amplification, especially using primer targeting V3 and V4. Some researchers, they prefer to go for V4 only. There's pros and cons for selecting either one, either both or only V4. And then after amplification, we do the sequencing. Mostly for our team, we use Illumina machine, and then finally the bioinformatics part. So this is the workflow after DNA extraction. The next step is fragmentation, and then after fragmentation, it's DNA sequencing. So after DNA sequencing, there are 3 important steps here: assembly, annotation, and binding. So for assembly, the reads assembled into contagious, and then finally into the whole genome, as you can see here. While for annotation, it involves identifying the protein coding regions, especially the ribosomal RNA. For binning, it's the sorting of the DNA based on sequence, based on the into groups, different groups. So here, this is the first group, second groups, second group, third group, into groups, which represent, each group represent individual types of genome. We call it here OTO, so this is how we do the taxonomic classification. Part of taxonomic classification or data analysis for 16S-RNA is the alpha and beta diversity. So alpha diversity is to see the diversity and abundance of the microorganisms within the same sample or within the same group of the treatment. And beta diversity is to compare between different groups or different samples. And also we study the richness and of taxa and distribution of the taxa as well. There are several bioinformatics. We use it for the 16S-RNA. So here I have listed few software packages that we use it for as processing tools. The most commonly used tools, Chung, and this is the website for that tool, or Dada2. There is other alternative, less commonly used like Motor and others, but the most researchers, they use Chung or Dada2. For taxonomy databases, the well-known SELFA or green genes, those are the most commonly used. Ribosomal database project is used to classify the microorganism. After I explained briefly about the metagenomics approach, let's move to the probiotics, because I will present some of findings by our team on the effect of certain or selected probiotics on the gut microbiota and also on the colorectal cancer. So what is the probiotics? Based on International Scientific Association of Probiotic and Prebiotic, it defined probiotics as live microorganisms, which when administered in adequate amount, it confer health benefits on the host. There are several studies that show that probiotics, it modulates first the gut microbiota, and by modulating the gut microbiota, it can play an important role in gastrointestinal diseases like ulcerative colitis or colorectal cancer. Many studies showed that lactobacillus, those bacteria or microbes that it produce lactic acid, we call it lactic acid bacteria, it counter or it inhibits the colorectal cancer. For the next slides, I'll show you some of the findings of 2 probiotics, Propionibacterium frondreshi and Faecalibacterium prausnitzii. Those some studies from the literature just to show that the probiotics, it has anti-proliferative in vitro against certain colorectal cancer cell lines, and also it modulates the immune system. It has apoptotic effect on colorectal cancer as well. And some studies, it showed that cocktail of lactobacillus and Faecalibacterium, so instead of having mono supplement, combining both, it produces synergistic effect. And this is the study that I have mentioned, that the clinical study that showed the intestinal gut microbiota in patients with CRC are different from the gut microbiota of healthy individuals. The first bacteria we used for our study, Propionibacterium frontreishi. It's from the family Propionibacteria, and it's safe because we use it normally as cheese ripening agent. While the second one is Faecalibacterium brusnesi. It's from the family Pharmacology, and it's oxygen sensitive. That's why for our experiment we use an aerobic chamber to grow this bacteria. And the good things about Faecalibacterium, it produces certain metabolites, short-chain fatty acid, including butyrate, which may have anti-inflammatory effect as well. So the aim of the study is to investigate the role of those two probiotics on modulating the gut microbiota in colorectal cancer model. This study includes 3 objectives. So the first objective, let me just check the time. So the first objective, I'll go fast. It seems I'm running out of time. So the first objective is to study the in vitro effect of those probiotics, while this using sample assay, MTT assay, while the second objective is to investigate the preventive effect of those probiotics against colorectal cancer in CRC rat models. So we induced colorectal cancer using carcinogenic azoxymethan by subcutaneous injection, and then we treated the rats for 6 weeks. And then, of course, to study the how those probiotics modulate the gut microbiota, we use the 16 rRNA sequencing, and those are the grouping of the animals, 6 groups. Normal control, the tumor control, 1 group treated with the Propionibacterium, the second group treated with Faecalibacterium, and then cocktail of both, and the last group as standard drug 5-fluorouracil. Each group contains 5 to 6 rats. And those are the results. Of course, we use an aerobic chamber to grow the probiotics, and the cell-free supernatant was freeze-dried using vacuum to be used for in vitro study, while the live microorganism used for the in vivo study. This is the result of MTT study. The findings showed that Faecalibacterium is more effective comparing to others, but of course it's not as potent as 5-fluorouracil. So the IC50 of Faecalibacterium, it's more much better compared to Propionibacterium. And also the MTT assay showed there is both concentration and time dependent. For the in vivo study, after we collected the colon, we have done macroscopic and microscopic investigation, and also the fecal material for 16S-RNA. For the body weight, the interesting result that the rats treated with 5-fluorouracil showed reduction or loss of body weight starting from week 3 until week 6, since the study for 6 weeks. For the macroscopic examination using methylene blue, the rats treated with carcinogenic azoxymethan showed apparent crypt foci, while here you can see the normal control, it showed normal crypt. The apparent crypt foci also observed in the treated groups, but with less. So if we see the table here, the percentage of inhibition, the 5-fluorouracil inhibit the apparent crypt foci, which considered as precancerous lesions, by 67%, while the second best inhibition is Propionibacterium frontreishi by 61%. This is just illustration for the same percentage of inhibition, and this is the histology of the histology result of the rats. So you can see here the normal architecture of crypt, while for rats treated with azoxystrobin, you can see hyperplasia and then elongated cells. There is infiltration of inflammatory cells here, while less in the treated groups. For the measurement of oxidative stress, since azoxystrobin, it induced apparent crypt foci by toxic metabolites, so there are oxidative stress. So as you can see here, there is the MDA is high in rats treated with azoxystrobin, and rats treated or supplemented with probiotics ameliorate or reduce this oxidative stress, especially Propionibacterium group. The last result is the effect of those selected probiotics on the diversity of gut microbiota. As you can see here, that Faecalibacterium, it increased the so here in the blue color Bacteroidata, it's higher in Faecalibacterium compared to azoxystrobin, the tumor control. So this is the quantitative. And the other strain, which is Firmicutes, it's less. Firmicutes, it's red color, so it's in cocktail and Faecalibacterium less compared to azoxystrobin tumor control. So the ratio, it's much lower. And this ratio, based on the previous study, it showed that the ratio of Firmicutes to Bacteroidetes, if it's lower, it indicates there is lower risk of colorectal cancer. This is the diversity at the genus level. As you can see in the probiotics groups, there is higher diversity, and there is higher of Lactobacillus in blue color, in Echinacea, pink color, and also it has, if you have a look at azoxystrobin, the tumor cancer, it has more green color, which is Escherichia coli and Clostridium and other strains associated with higher risk. This illustration, it shows the heat map, which reflect what I explained, the diversity and distribution of microorganisms at the genus level. So in conclusion, these findings, it prove that the consumption of certain probiotics, it ameliorate the effect of carcinogenic as a preventive tool, of course. And the inhibitory effect of Propionibacterium against apparent crypt foci is more prominent compared to Faecalibacterium. But on the other hand, the Faecalibacterium brusnesi was better in modulating the abundance and richness of the gut microbiota. But we need further studies to understand the mechanism, to do some certain metabolomics study and some proteomics study. And I think the second talk will focus more on the omics, the other types of omics. So with those some of the publication of this work published in the MG Open Journal, in Nutrition Review, and other journals. Here a list of the grants and some of the collaborators, and most of this work has been conducted by IFOMA and WARDA. And with that, I'd like to thank you all. And I have put my email in the first slide, so feel free if you are interested to collaborate with me, or if you have any question, feel free to contact me. Thank you, everyone, and have a good day.
Thank you so much, Professor Mohammed. Indeed, right, research on metagenome has been really exploding recently. I think also due to the increasing access and also decreasing cost of next NGS services. We also observe an increase in research output from other omics studies. I think we have in transcriptomics, metabolomics, and many more. So putting all this together, it is really daunting trying to make sense, right, from a healthcare point of view, how this omics data come together and what does it mean. So today,